right, ladies and gentlemen, we are back for part 2 of historical applications in biotechnology. When we last ended up, we were talking about Alexander Fleming and discovering penicillin in 1928, the same year that FFA was founded. So let's hop right back into the tick-tocky time machine and take a look at these further issues and applications in the history of biotechnology. So let's pick back up, skipping from 1928 into Rosalind Franklin and her contribution to the discovery of DNA. So in 1952, Rosalind Franklin, who was an incredible scientist at King's College, did early research using X-ray diffraction to learn about the structure of DNA. And what she did is she used X-rays to create images of these crystallized solids, okay? Now, this is a difficult concept to visualize, so I'm going to have a video later in the week or in the weeks to come that's going to explain Rosalind Franklin's contributions in greater detail, as well as her connection to two other individuals on this timeline. So right now, all I want you to know is that Rosalind Franklin was vital in discovering the structure of DNA. Now, DNA is deoxyribonucleic acid. It is the chemical substance that basically means that something is alive. Every living thing has DNA, and it was Rosalind Franklin's contributions here that very much helped prove the structure of DNA and how it was built. Now, this is combined with the efforts of these next two gentlemen, James Watson and Francis Crick. There's James Watson right there, and there's Francis Crick right there. And James Watson and Francis Crick in 1953, they revealed and published their structure of DNA. A lot of their work was honestly built on the work of one Rosalind Franklin. There's a whole story behind it that I'm going to share later. But between Rosalind Franklin and James Watson and Francis Crick, they were responsible for discovering the three-dimensional structure of DNA. Why is that important? Well, because we understand how the chemical of DNA is built, how it's structurally held together, and the way it also told us how DNA copies itself. And so we learned, based on understanding how DNA is built, how DNA, well, continues to exist, which also helps us understand how genes and genetic traits are passed from parents to offspring. So this is a huge deal. Now, later on, there's going to be a whole video that you guys are going to watch and have talking about the work between Rosalind Franklin and James Watson and Francis Crick, and this whole issue between the two of them. But right now, what you need to know, 1952, Rosalind Franklin produced early data about the structure of DNA, and James Watson and Francis Crick in 1953, they revealed the three-dimensional structure of DNA. Now, moving forward into the 70s, this is Dr. Norman Borlaug. And we talked about Dr. Norman Borlaug very briefly in the 4 areas of biotechnology video in our previous lessons. Dr. Norman Borlaug was an agricultural researcher and a pioneer in feeding the world's population. And in 1970, he was awarded the Nobel Peace Prize for his efforts in basically hybridization research and early genetic research to create varieties of wheat and corn that could be grown in very dry and complex conditions. So Dr. Norman Borlaug was born in Iowa in 1914, and he grew up on his father's farm, and he attended a one-room schoolhouse for 8 years. And he went on to college and got his bachelor's of science in forestry and his master's degree in plant pathology, which is the study of plant diseases. And then he got his PhD in plant pathology and genetics. So basically, he knew what made plants sick and how their DNA worked, how their genes are passed from one generation to the next. So he graduates from the University of Minnesota and then takes a job with the DuPont company. And for 30 years, Dr. Norman Borlaug worked as an agricultural researcher, and his efforts focused on improving wheat. Dr. Borlaug won the Nobel Prize for Peace in 1970 in recognition for his work in improving the living conditions for millions of people in developing countries, and he's referred to as the father of the Green Revolution. And he also continued his work until his passing in 2009, working in Africa to battle against starvation, genetically trying to get countries to adopt genetically engineered corn seed. Now, we talked about genetic engineering a little bit, but here are our OGs, and I'm missing photos of them, but we'll see photos of them later in our notable people presentation. So Stanley Cohen and Herbert Boyer in 1972, they performed the first successful recombinant DNA experiment using bacterial genes. Well, what is recombinant DNA? Well, let's take a look at our picture here. So recombinant DNA is basically when you take this little strip of DNA right here. This is called a plasmid. Bacteria make these circles of DNA called plasmids. And what we do is we literally cut that circle of DNA apart and we add a new gene that we want. So for example, a gene for human growth hormone. We can add this gene, paste that gene into it, and the DNA becomes recombined, recombinant DNA. So basically, it's taking a small strip of DNA, separating it, cutting it apart, adding a new link of DNA, and completing the circle again. And so it has a new trait, new genes that we want to have. And then we place this recombined DNA back into a bacterial cell to grow a whole bunch of those plasmids and a whole bunch of those bacteria that have that new trait. So recombinant DNA is essentially genetics engineering's most basic form, taking a trait from one living thing and placing it into the DNA, the genetic code of another. And so Stanley Cohen and Herbert Boyer, they were scientists, and they got together and partnered up to begin their work in genetic engineering after they met at a conference in 1972. Then in 1973, they invented the technique of DNA cloning called recombinant DNA. And this allowed genes to be transferred between two different biological species. As a result, they are responsible for genetic engineering. It was their work that opened the door for biotechnology. And in 1976, they founded the company Genentech, which would be a world leader in biotechnology. So, this is an important one in the history of biotechnology. So, I'm going to show you the image first. So, this is Dr. Ananda Mohan Chakrabarty. And he created a bacteria that would eat oil. Okay? Basically, eat leftover oil that would be broken down into harmless components. Well, he was working for a company called General Electric, GE. And they, along with Dr. Ananda Chakrabarty, patented that gene, the gene that changed this bacteria to an oil-eating bacteria. Well, there are some folks that were not happy that General Electric and Dr. Ananda Chakrabarty patented something that they felt was a natural thing, a living thing in a bacteria. So they sued him. Well, this went all the way to the Supreme Court. And so when in 1980, Dr. Ananda Chakrabarty won his Supreme Court case against the Sidney A. Diamond, the Commissioner for Patent and Trademarks, and this meant that from here on out, the precedent was set that companies and researchers could be granted patents on novel genes, genes that they created and added to bacteria. This allowed for biotech companies to be able to own their intellectual property. That meant that they would be able to create medicines, be able to create new living organisms, and they would be able to own the intellectual property and technique of how that organism was created. That is very important because this led to dozens of biotechnology companies being founded for many, many different reasons. And so these different companies brought different ideas to the landscape of the biotech revolution and led to advances in agricultural biotechnology, food and consumer biotechnology, environmental biotechnology, and medical biotechnology. Now, another thing that is important about biotechnology is that it also is used to understand how diseases work. Before 1981, acquired immune deficiency syndrome, or AIDS, and HIV was considered a form of cancer. No one really knew how it was transmitted. And in 1981, because of tests created using biotechnology, AIDS was identified as a virus, a communicable virus that was passed from person to person. This led eventually in the future to treatments for the HIV and AIDS virus, turning something that was a death sentence to something that was more survivable. Now, moving forward, 1982. So, guys, there are a lot of individuals who suffer from the genetic disorder diabetes. Diabetes is when a person's body and pancreas no longer produces insulin, or it produces too much insulin. So what happens is, in order to treat this disease, Genentech, remember, going back to Stanley Cohen and Herbert Boyer, that was the company that they founded, and the first thing that they attempted to create a treatment for was diabetes. And so Humulin was a genetically engineered insulin treatment, and it was the first biotech drug approved by the Food and Drug Administration in 1982. And Humulin was Genentech's human insulin product produced by genetically engineered bacteria for the treatment of diabetes. And it is vital to understand it was the first of many biotech created drugs that were approved by the FDA. All right, so this is Robert Hirsch, and he was basically a plant tissue expert who dabbled in some genetic engineering, and he figured out that bacteria, particularly a soil bacteria called Agrobacterium, could be used to genetically engineer plants. Essentially, what it does is you put a genetically engineered agrobacterium with the trait that you want on it. For example, we want to put a BT or Bacillus thuringiensis protein in a plant. Bacillus thuringiensis is a soil bacteria that if caterpillar and other larvae eat it, it basically kills them. It makes it so that their stomach the bacteria makes it so that they can no longer get nourishment from what they eat. And so the bacteria eat until their internal organs basically. And so it really doesn't take that much if the insect isn't digesting, if they're not getting nourishment from the food. And so Hirsch was a genetic engineer who did several experiments that proved that Agrobacterium could genetically engineer plants. And this was a big step forward, especially in agricultural biotechnology. Now we could pass genetic traits from one species to another, not just in bacteria, but in plants as well. And that was the responsibility of Robert Hirsch in 1982. Now, in 1984, Dr. Alec Jeffreys, a geneticist at the University of Leicester in Great Britain, who was studying hereditary diseases, he began to focus on how you could prove people were related to one another. And he developed this technique that is used called DNA fingerprinting. And basically what it does is it allows an individual to run DNA sequences and compare them. And the closer a DNA sequence is to one another, the more closely related those individuals could be. And so DNA fingerprinting is now used as a way to match organ donors. It's a way to solve crimes. It is a way to discover paternity of and find long lost family members. And so DNA fingerprinting was discovered and developed in 1984. Taking it into 1987. So, back in 1987, Dr. Ben Carson was the first surgeon to successfully separate Siamese twins who were joined at the head. Dr. Carson was a surgical genius, and he focused on pediatric neurosurgery. That's a fancy way of basically calling him a brain surgeon for, well, babies and toddlers. And so Ben Carson grew up in inner city Detroit and Boston, and his parents, they separated and were divorced when he was about 8 years old, and he and his older brother were raised by their mom. And Ben struggled as a kid with failing grades and low self-esteem. He had behavior and anger problems. And in school, about middle school, Ben started to turn himself around. And he eventually got a scholarship to Yale University and continued his studies to the University of Michigan Medical School, where he studied neurosurgery or brain surgery. After he graduated, he moved to Johns Hopkins University Hospital, where he worked his way up as a surgeon and became the director of pediatric surgery at the age of 32. Dr. Carson gained worldwide recognition in 1987 when he became the first surgeon to separate conjoined twins who were joined at the head, a feat that had never happened before. Now, the Human Genome Project. So, in 1988, with the development of DNA fingerprinting, Congress decided to put that development to work and fund the Human Genome Project, which was a huge effort to map the human DNA genetic code so that we could understand all of the genomes and the DNA structure of the human species. That way we could potentially find out treatments for genetic disorders and potentially find cures for diseases that we think are uncurable, like cancers. And so the Human Genome Project was completed in 2001. We now have an entire length of our genetic code uncoded. And now we're trying to understand what each of those genes do. And we have a huge database that can find, and if people are interested, to look up what different genes do. And this is used by helping doctors figure out how to treat patients with different genetic disorders. Now, 1990, there's a 4-year-old girl named Ashanti da Silva, and she had a genetic disorder called ADA-SCID, a severe immune system deficiency. This meant basically that she had to live in a bubble. She had to live in a plastic dome, and she could not go outside due to potential interaction with, viruses and bacteria and all the things that would make us sick. In 1990, the FDA approved a gene therapy to treat Ashanti da Silva, and the effects were temporary but successful. This led to the creation of many more gene therapies that help improve the quality of life for thousands of people all over the world, individuals suffering from many different types of genetic disorders, like this immune disorder that Ashanti was suffering from. And we're missing the picture of Dr. Ian Wilmot, but we do have a picture of Dolly the Sheep. So, in 1997, Dr. Ian Wilmot of Scotland was credited with cloning Dolly the Sheep from adult sheep cells. Now, this is important, okay? Because never before was there an animal that was cloned from an adult mammal. We'd done it with frogs, we'd done it with lizards, but we had never done it with an adult mammal, warm-blooded. And so we're going to talk about in detail the process of cloning at a later time. But Dr. Ian Wilmot was credited with cloning Dolly from adult sheep cells in 1997. This is Polly, the donor mother, and this is Dolly right here. These two are genetically identical. They are clones. And ending our presentation today. I know it's a few minutes over. In 2001, President Bush decides to permit federal funding of research using existing stem cells. So stem cells are these cells that basically are blank. They don't have any real, well, they have a genetic code, but it's open. It can be anything. If you put stem cells next to skin cells, the stem cells will become skin cells and create skin. If you put stem cells next to nerve cells and brain cells, the stem cells will adopt the characteristics of the brain cells and the nerve cells and become brain cells and nerve cells. So this is important because stem cells could be the future of treatments for diseases like Alzheimer's, Parkinson's disease, and other neuromuscular disorders. So, guys, this ends our presentation on the historical applications of biotechnology. There will be a timeline posted in Canvas for you guys to go back and look. Also, the Quizlet for this week is going to be on the dates and times of this timeline. I'm also going to put in the TikToky timeline for your information so that you'll be able to go through, browse through it, and also maybe go and watch some of the videos, read some of the finer details that I may not have covered. And essentially, guys, that's this. Enjoy the lesson. I'll talk to you later.